I normally do a lot of RNA work and stuff like that, but at the moment I'm finishing up for a couple of experiments for a paper, so I thought that that would be a bit more interesting. I'm also doing all of what Jack just presented, so I'm not mentioning that whatsoever because I hate it. I've only done it for about 2 months, so I prefer the whole RNA side of things. But I'm going to instead I've been looking at lucky enough to get tissue samples from patients, so clinical samples. So I'm just going to, that sounds really, really amazing, and it is a great opportunity, but I'm just going to explain what we actually do with them. So, my project is also, I basically decided in order to tell you what I'm doing with the samples and why I need to do what I do with the samples, I need to actually explain a teeny tiny bit about what I'm actually studying, because it makes a bit more sense. So I'm studying inflammation, specifically around rheumatoid arthritis, but inflammation is a very common thing. You know, if you have a cold, like certain people in the audience, that basically, you know, the inflammation, there's a surge of inflammatory cells which then give you a fever which fights off the bacteria or the virus or whatever that's happening, and it basically resolves and then you feel better again. And in a lot of diseases like this, like in autoimmune diseases, arthritis and this kind of thing, the inflammation is a lot more sturdy, and it can last for a lot longer, so it's called chronic inflammation. And so basically, I'm looking at rheumatoid arthritis, which everybody kind of knows is a chronic inflammation of joints and surrounding tissues. It's an autoimmune disease, and so the body's immune system basically thinks that the healthy tissue is actually a disease. It's trying to get rid of it, and so it attacks it basically. And this happens in a lot of autoimmune diseases. And the statistics are roughly 1 in 5 people in Ireland are going to have some form of arthritis, not necessarily rheumatoid, but some form of arthritis in their lives. And most people, they won't get rheumatoid arthritis until they're about, say, in their 40s, but there are other forms, which is called juvenile arthritis, where people in their young teens or younger can actually develop arthritis, so they can be dealing with this disease for their whole lives. So it's very important that you get as much information as possible about a disease so that you can go about curing it, making drugs, to help with the condition or even cope with the condition. So you're forgetting I have a pointer. So what happens in a joint? Basically, this is the 2 bones, so I think this is a knee that they're trying to show. So you have your bone here and here, and you have your synovial space in between. Everybody kind of knows what a bone is supposed to look like. And you have, you know, your ligaments and stuff holding things together. And you have this really thin layer of cartilage right here. Can anybody actually see the pointer? Yeah. Otherwise I'm just pointing at random space. So there's this layer of cartilage which is supposed to be like a cushion or a buffer on the edge of your bone, so that if they end up like, basically it protects the bone from being degraded further back into the bone, or being eaten away or rubbed against each other. And what happens in arthritis is, well, this is osteoarthritis, and is that the synovial fluid basically is completely depleted from the bone, which and the cartilage is eroded away, which means that the 2 bones are basically rubbing against each other, which is what the pain is caused by. The pain is just the bones going like this. And what you need to, and in rheumatoid arthritis, what happens is the cells get a bit confused. And it's kind of a bit like kind of cancer in a way, and that the cells they basically form this thing called a pannus, which is a cancer-like membrane, I suppose, around the bone, which means that the bones, in this case, the cartilage is depleted, and in this case it's depleted, but it's actually replaced by extra bone, which means that the bones technically grow into each other. So that's what happens in this, and also the synovial fluid kind of is depleted, which means that there's less cushioning involved in this. So, what else would I say about this? So that's basically what the difference between normal and rheumatoid arthritis is, what I normally study. So just to show how quickly this can occur, this is a normal joint here, and 2 years on you can see that the space between the bones indicated by the arrow is completely depleted, and by year 3 the bones are practically grown into each other. So this is really, really progressive. Now this would be an aggressive form of arthritis, but still it can happen really quickly, so you need to know you need to be able to diagnose someone with arthritis as soon as possible. So I'm going to just use this because I use this image all the time, so I know it back to front. But I just want to point out basically, so just in a little cartoony form, this is the bone, here's the layer of cartilage here, and what you'll have is you'll have the synovial space in between here. Now what happens is that the cells are, there's some form of stimulus, be it stress or some factor, genetic factor, that causes the release of cytokines into this into the synovial space in between. So cytokines are like, if you have hay fever and you have a histamine response, you take antihistamines for it to dampen the effect of histamine. So these, mainly the two huge factors known in arthritis are TNF-alpha and IL-1 beta, and there's drugs against both of these factors. So basically, you have the stimulus, it tells the cells to start producing these factors, they're inflammatory, and in a normal circumstance, if you had a cold or a flu, they would signal, they would be in the space, and then they would target the bad cells, so to speak, like the bacteria, kill them off, and then they'd deplete. They'd kind of send a message to the rest of your body saying, okay, done that now, it's all better, and they'll go away. Whereas in arthritis, that message is kind of lost. So instead, these factors cause proliferation. So what was shown in Jack's slides with the cells dividing, it basically signals to all the cells in your bone to keep producing bone cells. So it kind of says, no, all these cells are fine. So when the cells should be dying because they're a bit crap now, they've reached the end of their life, and they should be naturally degraded to replaced by new cells, because your whole body replaces itself and replenishes itself over time anyway. It's that instead signals to the cells going, nope, these are all fine, keeps them, which means you have dodgy cells growing, and this is this kind of cancer-like panis that starts to grow. So the bone starts to invade the space, grow over the cartilage, and instead you'll have bone cells which are, well, reached the end of their life, starting to creep up into this space here. So that's kind of the premise of rheumatoid arthritis. So this looks really complicated, but it's a really simple way of doing it. So what happens is, just to reiterate what I just said, is that you have inflammation normally, and then the cells rectify the situation, and then it goes into this resolution or tissue repair stage. Everything gets fixed, and yeah, the body goes back to normal, doing its normal everyday things. In chronic inflammation, the difference is that this resolution stage, this kind of dropping away of these inflammatory cells, never happens. So instead it continues on. Which means there's lots of proliferation of these cells, so loads and loads of cells get formed, and nothing gets regulated, and when you don't regulate genes, they can just go completely out of control. And that, what I'm studying is this little red bit in the middle, which is basically what I wanted to say about it is arthritis. It affects 1 in 5 people in Ireland, but if you diagnosed those people in the first year, because of that bones picture I showed a few minutes ago, if you diagnose them in a relatively short period of time at the start of when they're just starting to become inflamed, with the drugs that are currently on the market, 80% of people will go into resolution and the arthritis will be completely cured. So, but unfortunately, people generally don't go to the doctor with symptoms until their joints are already sore, they're already swollen, they're already kind of past the point. So then the drugs become a coping mechanism, dealing with pain, trying to prevent further degradation of the bones, but they won't actually be able to go into this resolution. Like, I suppose arthritis, you hear about the kind of peaks and troughs, so people will be really highly inflamed and they'll be fine for a little bit and then they'll be really highly inflamed again, but that basically inflammation will occur throughout their whole life. The drugs will just be a coping mechanism. That makes sense? Whereas if you can catch them in an early phase, it will actually be a cure. So there's a huge importance in actually finding information about this bit, what actually happens here, because it's not actually known, rather than the later stages, so that you can actually help people in future even with the things that are on the market at the moment. So what we want to see is look at that target area, that early stage of inflammation, what actually happens, what spirals out of control to cause the arthritis in the first place. so the physiological effects of different factors at that stage. And we also want to find targets, so maybe, okay, that the drugs on the market only affect 80%, would help 80% of people, that's a huge amount of people. But the other 20%, actually none of the drugs on the market will actually do anything for. They're completely unresponsive, and unfortunately it's not helping their symptoms or helping their pain in any way, shape or form, so they're just on painkillers the whole time. So you need to be able to, at some stage, to design drugs targeted for each person, so you can do like a blood test. You can go, oh, you have this particular type of gene expression, and we need to give you this drug that'll make everything better. So to be able to design specific drugs, you also need more information as well. So, so how do we go about achieving this? So this is why I'm working with patient samples at the moment. So we do sometimes get arthritis samples from arthritic patients, and that is fantastic to get them, but the issue with that is because they've gone through so much degradation, there's very little cartilage or bone left. Like there's the unusual cells, but there's none of the normal cells left there for you to be able to analyze. So, and it tells you stuff about the end, or like say 6 years on, or 20 years on, or 50 years on into the disease, but it doesn't actually tell you about those initial gene expression events that are happening that cause it in the first place. So we want to have healthy joints. Now you can't just ask people to kind of walk off the street and give you their joints to use. So what happens actually, how they're deemed healthy, is basically people are going in for hip replacements, but they're actually going in for hip replacement because they've been in a car accident, or they've fallen down the stairs, or they've tripped, or they've shattered their bone, or something like that. Some event that has happened that means that they have to have a hip replacement, but otherwise they're perfectly healthy people. So this does happen from time to time, and they had a little bit of a joke about it in the hospital, saying that last year there was a lot of ice, and they were kind of looking at the window at the car park, waiting for samples because there was someone just finishing off their PhD. I know I shouldn't say I can see you, but someone was just finishing up the end of their PhD and they really need one more sample. But no, these are actually normally, they're all emergency surgeries, so I can get a call at any time during the day or evening, because they generally don't work in the night, thank God, or the weekend for these samples. So I'm working with St. Vincent's, which is the only hospital in Ireland that will actually give these patient samples. All they have to do is toss the bone when they've taken it out of the patient into a bucket and hand it to a research person. That's all they have to do. They don't do anything else about it. Every other hospital throws it into an incinerator and burns it instead. So this is the only hospital that actually kind of actively helps out research projects. So basically you can see I get the femoral bone. It's basically the hip bone, ball and socket joint that's at your hip. I get that, and so basically you can see that there's this little, in a normal joint, there's this lovely kind of coating, pink coating of cartilage on the edges here, whereas this one is degraded, so you can see that the bone is leaking through in this one. So we would use this bone. You can't actually use any of the degraded, you have to use over here, whereas this one you can use anything from here. So the healthier the better, really, for our experiments. So the first step is to wait on a call from the hospital. I was waiting 2.5 to 3 months for the second one, so you need a couple of different patients because there's patient variation. And then you have to harvest the cartilage. Now, I'm not actually going to show a picture of this because it's a little bit gross. So this is him carving a ducky instead. But it's the same principle, really. I have to put the chainmail on. I have to use sterile tissue culture like Jack was talking about, and you basically have to, with a scalpel, shave off the cartilage layer, the top layer of the bone, because the bone isn't what's important, it's actually the cartilage what we're looking at. So the ducky just is a nicer version rather than the bloody specimen. So then we have these like little punches, which if anybody's got a tunnel or any other kind of stuff, they use the exact same thing. And basically this would be my little petri dish here. You have all the little slices of the edge of the cartilage, and you can see all the little holes that are taken from the punch. So this is, you keep them immediate to keep them nice and healthy, but that's basically how you go about taking the samples. Now, what I can also do from these is the edge bits, like that the holes haven't been taken from. I can also digest them with enzymes and make primary cell lines from that as well. So I can actually take them from the patient, start making them into cells, because this has a lot of collagen and stuff holding them together. Take away the collagen, basically, grow them in how traditionally should grow. They take a few weeks though, so I'm still waiting for one to actually wake up a little bit. But so then I put them in a 96-well plate, and so you can see I have them all labelled. This is my first plate when I was being really, really pretty about my labelling. The rest of them aren't actually labelled at all, they're just kind of roughly boxed. But you can see that there's a little kind of orangey pink media in them, so that has your, we actually use an antifungal kind of, as well as all the other antibiotics as well, because it has been thrown in a bucket and then given to us. You never know what was in the bucket beforehand, it's better just keeping everything clean. And what I have to do is I feed them, which sounds like a really lovely thing, it isn't pet, every 4 days for 3 weeks, so per patient. So you need a couple of 4 or 5 patients probably to be able to have publishable results, and that's if they're all within a margin of error, so to speak. So I just said that. So there's my nice big plate. I was trying to demonstrate how small they are, so you can kind of see here that the plate is about a little bit bigger than my hand, not quite bigger than my hand. Then you can see that there's little teeny tiny little circles. Pictures are taken on my phone, so they're kind of small. But I wanted to show that there's a 1 cent coin. That's how small they are. They are really finicky to play with. I decided to play with it then after that. So that's how teeny tiny they are. That is a really tiny tweezers. And basically they're as fiddly as shit to work with as well. Because you have to place them into the individual wells with the tweezers. Then you have to feed them, make sure they don't go up your pipette tip when you're feeding them. Sometimes they kind of degrade a little bit and they suck up as well, so you have to put them back in again. And then afterwards you go through a lot of things which I'll talk about in a minute. I'm getting ahead of myself. I get really excited about the carbon. You've talked to me recently, I talk about this all the time. But yeah, so it's 3 weeks. So under experimental conditions basically means that I'll put my different antagonists, my different drugs, my different proteins of interest, whatever, into the different wells in triplicate, because that's scientific. Basically to treat them with different things to see which one, well, what does what, I suppose. What I was actually trying to prove for the paper is that a particular thing that we're looking at makes them worse. So we're trying to show that this causes arthritis. So basically we're taking healthy tissue, pouring cytokines on it, and then basically going, look, see, it's worse now. So that you can, if that is true, we can go back, we can tell other groups to design drugs to block it at the first step when it's still healthy tissue, so that it won't progress to getting worse, if that makes sense. So we use TNF-alpha as our positive control. So basically, the most common anti-arthritis drug, or treatment for arthritis, is an anti-TNF-alpha, which basically blocks the action of TNF-alpha in the joint, which then basically stops it from getting any worse, really, for most people. So we're using it as a positive control, so we're testing our reagent in comparison to the known one that's quite potent. So it's 4 days for 21 days is one experiment. And then after, what we do is you have to pick up each piece of cartilage, and you have to fix it in formalin, so basically formaldehyde. And you have to make sure that the cartilage doesn't degrade anymore. Because you've just basically been keeping it healthy and alive for 3 weeks once it's been out of the patient, and then you have to put it in formaldehyde basically to make sure that everybody's seen formaldehyde samples in like one of the National History Museums or whatever. They stay for a very long time. It means that nothing gets worse than what it is at the moment. So it's very easy to dunk it in. It's not that big of a deal, but it's just the amount of samples that kind of jumped across the table because you've kind of gripped them with the tweezers and they've gone flying across the room. So then the next stage is embedding. So what they're doing here is they're embedding the sample in wax. It's a paraffin wax. And basically, in order to do this, what they need to do is go through a whole load of stages of dehydration. So what you're trying to do to the sample is basically the wax and water mix together, and they basically, it makes the wax a bit watery, so the wax won't set properly. So what you need to do is basically put different levels of ethanol. So you start with 70%, then 80%, then 90%, then 100%, until it gets all the water out of the sample. So it's fully dried out. And then you put xylene in, which just basically takes the place so there's no air bubbles in it. And then the wax basically kicks the xylene out and embeds into the sample. And that means that you can slice the sample really, really thin to make microscope slides. So what she's doing here is basically holding the piece of cartilage into the wax on its side until it gets dry. So you have to basically wait there with the thing, with the piece of tongs, waiting until you basically put it on a cold bath to try and cool it down quicker. That's how finicky it is. And then I have a video which I need to check out for And everybody 3 minutes long? So this bit I'm looking up at someone else is doing for me because I had a deadline next month, so I haven't got enough time to do this or learn how to do this. And you can see how tricky it is when you actually see it. So what they're doing is, here's a sample, they're putting it in the machine, and it says underneath, I'm just going to talk over it anyway. You basically need to, so you'll have your sample peeking it. You don't want to use the bit that's closest to the edge. You want to kind of cut in a little bit so they're all rectangular, so they're all kind of the same size. So that's, you just kind of cut a bit off, a chunk off at the edge first, and then you basically turn it down to 3 to 5 micro. My color is it was a UN, so I can't remember what it stands for. And yes, basically, so it's really, really thin, so you can see tiny thin sheets of wax and sample that she's cutting here. So I know I will be terrible at this when I actually have to learn how to do it, because it looks even more tedious than thinking. But yes, you stretch it on water to kind of stretch the sample out. If there's any folding in it, then the sample can't be used really because it'll interfere with the images that you get at the end. And then you catch it on a slide and try it. So it's, I don't know, I have quite a number of samples at the moment to process, so me even trying to do one of these would probably take about 10 hours because I'd be trying to catch it. But that's, and then basically all they do, there's no glue, all they do is basically wait for the slightly warmed wax to stick to the glass slide at the end once they've caught it at the end. So it is quite, it looks really scary for me to do, but I will have to do that, learn how to do that the next couple of weeks. So then the next stage is staining. So you have your staining buckets with your different dyes in it, and you have, these are the staining slides at the end. So what I've been doing is staining my cartilage with 2 reagents, saffron and O and fast green. And basically this is a test to look at gangrenous is what we call it, but I can't actually pronounce the full word. So it's proteoglycans, which are basically the components of like the extracellular membrane. Basically they keep the cells intact, and so the more it's lost, the more degraded your sample is. So if they're nice and healthy, there won't be any, only the red stain, the saffron and O will stain the sample, so they'll all be red, they'll be looking healthy. And if you have any of this fast green, it means that your sample has been degraded and you'll see the level of degradation. So that's kind of the principle of what we're doing with the stain slides. So this is kind of what it would look like in a particularly bad sample, is you have the red here. We have a hematoxin, which is a nuclear stain, so you can see the teeny tiny blue dots from the nuclei. And so this is all good tissue here. You can see all here is green, so this is quite level. You measure the distance between here of being particularly degraded cartilage, and then the bit in the middle is kind of ebbing in, so it's degrading back further. So this would kind of be, this is cartilage sample, so it's the layer covering the bone. So this is the amount of proteoglycan lost, so the amount of degradation that's happened to this piece of cartilage is out back that far. So just to kind of look at what we would look at in a paper, it would be, this is control sample. So there's a tiny little bit at the edge, but nothing major. There's a tiny diffuse streak, but everything else is really red, really pumpy red. They all kind of look a bit like watermelon, I know that. So this is your TNF alpha, which is your positive control, and kind of this would normally be present in arthritis and tested for. So you can see there's quite a distance here seen of degradation. Then this is the factor that we're looking at. There's a little bit of degradation happening, or it's about the same thickness as here, but it's not quite as green. But when you add the two together, it's quite double the distance back. So they're additive effects. So they both have an effect on the cartilage, and when you add them together, which would happen in a normal joint, because you have you don't just have one thing. You don't just have TNF alpha. It doesn't pick one and then stick with that. It has loads of different factors being sucked in. So this would have an additive effect and make the cartilage even worse. So I just wanted to point out, and this is where I stopped. This is my last slide because I got a little bit freaked out. So I've gotten 6 patient hip joints from the hospital. So that means I have 219 pieces of cartilage, circles of cartilage, to be able to section them, mount them on slides and stain them and image them. Then I have 87 samples that I'm going to do RNA extraction on later, so I'll be basically getting a mortar and pestle, grinding them up, and then extracting or testing them for gene expression of different markers in these samples. I'm not going to go into that because I'm not going to be doing it for a few months. And then I have 306. That adds up to 306, I hope, because I was doing this quite hard. Cartilage samples that were fed every 4 days for 21 days. Now, when I fed them, I had to keep the supernatants because basically, if these factors are being released, they're being released into the media. So you can test the media itself as well to see whether they're being kicked off, which means that I have 5 collections for each one. So I have 1,836 individual tubes of supernatants to analyze. Then you have to do duplicates, which is 2,672. And that's if everything works right first time. And if not, then I have to kind of pipette these 3,672 samples over and over again. But basically, that assay is going to consist of, you pipette the sample in, it's a really fantastic assay. Pipette the sample into another 96 well plate, and then you put a blue dye in it, which basically it deposits based on the amount of proteoglycans that have been released into the supernatant. So the darker it goes, the more that's been released. So there's another control. So you'll have the images, you'll have an assay with a nice little graph. I'll have RNA eventually. So from those few patients, I have kind of generated that much. You should see the boxes and boxes of supernatants I have at the moment. It kind of is about the same size as me. So I have a lot of work to do. So I'm a little freaked out. But yeah, basically this is the importance of patient samples from these few patients. There's only 6 patients, okay, it's quite a lot. But 6 patients worth of samples I can do my analysis of my ELISAs, I have all my imaging, I have RNA, I'll also be asking for permission, ethical permission to ask whether any of these patients were actually on different drugs, whether they were on anti-TNF alpha treatments or anything, so I can match them up to my results to see whether these patients are to do with drugs or not. So I'll have all that extra information. But there is a lot of ethical, there's a lot of red tape. So I just get a random arbitrary number, but they don't even tell me whether the patient's male or female most of the time. So it's kind of, you just get a random sample thrown at you. But yeah, that's basically what I'm doing at the moment. So it's a lot of work, a lot of long hours, and a lot of petting. So if you have any questions, let me know.
Permission, or was it that your lab already had permission?
My lab already had permission for most of it. Basically, what we're doing is having collaboration with the hospital, so they already had ethical permission to use whatever samples they want. And the permission is actually written on every single consent form when you're going in for an operation. It's in the kind of small, not quite small print, but it's in one of the sections that people don't normally read because they're going in for an operation. And that they basically said if people need this for research and it's not needed for any other purposes, then it would be given for research if that's okay and you've signed off on it. The only reason it wouldn't be given for research is if either A, the doctor forgets about it and throws it in the bin, or B, if there was any concern about the bone, if they went, oh, this patient hasn't been diagnosed with arthritis but it looks a little bit, and they send them off for actual beneficial results for the patient themselves. But the ethics, they can kind of, thankfully, I just have to kind of read everything through, sign off on it, promise that like you have to go through a lot of rigmarole. You have to, you're not like, even my arbitrary number said I got 4527. I just picked those numbers out of my head. That was not one of my patient samples, just like but if you get that and you write it even on a plastic bag with my super names on it, when I'm throwing the bag out, I have to cross it out and cut it up, even though it only relates to an arbitrary number that they give me, not actually the patient sample. Like you have to, when you get the patient sample, it's in a little tube, you have to scratch off everything to make sure that there's no possible way of them being able to trace it back. So there is a lot of like legal stuff that you have to do for that reason. I'm also had to apply for separate ethics to be able to bring the samples from the hospital to UCD, because I'm based in UCD. So I know it's only a 10-minute walk, but because I'm technically carrying pieces of patient out of the hospital, I need to, like, they need to know where they're going, what they're going to be used for as well. I know I'm not very good at phrasing things, but yeah, so there's thankfully a lot of it was already pre-approved, because we also have animal models coming up in the next few months as well, so we've already got an ethical approval for that, and already got an ethical approval for those other things, so I don't really remember. I know what I have to do rather than the specifics.
Is there a particular reason that no other hospitals would participate in that?
I think they just think it's a little bit of work, because technically it's easier if they have a research department in the hospital, because what we're actually doing is, I have a lovely friend in the hospital who basically gives me a text every time that there's a person going into the operation theatre. You need someone to do that, otherwise, like, how would you know that they're going? If they're all in emergency, they're not scheduled, so they're not like, oh, there'll be one next Tuesday. There's one in half an hour, get your ass down here. So you kind of drop everything and run down. So that's mainly the reason, is that they'll have to get one of the nurses, or because the surgeons will never do it, one of the nurses or someone to phone you to tell you to come pick it up. And if happen to be in Cork, then they have to ship it to you, and they have to put it, so there's a little bit of effort there, and that's the only reason.
So it's not like really illegal or an ethical
No,
thing?
absolutely nothing. They just couldn't be bothered.
Yeah, right.
So there's a lot of us actually that again, patient samples. There's another group in the next lab that are doing colorectal cancer, so they get a lot of, when there's colonoscopies and stuff like that, they'll get samples from that too. It's just a matter of asking, being really nice to one of the nurses, generally one of the head nurses, and asking, giving them your number and asking them when they want it. That's it.
More to do with like having a working relationship with